Welcome to the NEC Society's webinar on basic science and clinical NEC research. We're going to go ahead and get started because we have a lot of content we want to cover. So welcome. I want to quickly acknowledge our partners. We are thankful to Engage, Grow, Thrive for partnering with us on this webinar, as well as Pediatric Research. And we keep babies and families centered in everything that we do here at the NEC Society. And so I'm really grateful to dedicate this webinar to baby Tom. Tom is who you see here in these beautiful photos. Tom's aunt, Simone, is the founder and director of a nonprofit charity organization that's based in Brazil. And I believe my colleague Erin is going to share a link to PGG, the charity organization based in Brazil, in the chat box. So if you're curious to learn more about that charity, you can click that link. But Tom developed necrotizing enterocolitis after being born prematurely. Tom is actually a twin. And Tom survived initially his bout with necrotizing enterocolitis and was doing well. And it looked like he was going to be okay and live a relatively healthy, normal life. But he had complications from NEC, and tragically, he died from those complications. So his twin brother is doing well today, and we carry Tom's beautiful smile and memory in our hearts and in everything that we do. So if you can keep Tom in your mind and in your hearts today, and as you continue on to do this work, that would be wonderful. But again, I just want to say thank you to Tom's family for sharing his beautiful face and smile with us. It's always just an honor to remember Tom and carry him with us. I'm going to advance to the next slide and get us started. So for anyone who is just learning about the NEC Society, welcome. We're so thrilled to have you. The NEC Society is a 501c3 nonprofit charity organization that is dedicated to building a world without necrotizing enterocolitis. We bring together patient families who have been personally affected by the disease, along with clinicians and researchers who have dedicated their entire careers to ending NEC, and other stakeholders who also care and want to help us advance education, advocacy, and research. So here's some of our team members here from pre-pandemic times when we were able to get together in person, and hopefully we'll be able to do that again at some point in the not too distant future. If you would like to learn more about what the NEC Society does and some of our proudest achievements, you can check out our accomplishments page. And again, my wonderful colleague Erin is going to share that link in the chat box if you'd like to learn more about the research, the education, the advocacy work, and other projects that we do, and how you might be able to help us raise awareness and advance this work and build a world without NEC. You can check out that link and visit our website to get involved. So here are your speakers for today. I'm not going to give introductions to everyone because that's going to happen a little bit later, but I do want to introduce myself. I'm Jennifer Canvasser. I'm the founder and the executive director of the NEC Society. I founded the NEC Society after my son Micah died from complications of necrotizing enterocolitis just before his first birthday. And just like many of us here at the NEC Society, we're really compelled to help other families not experience the devastation and the trauma that we experienced after our baby's diagnosis with NEC and after we lost our child to this really devastating disease. So I'm really grateful to our team here who helps make this work possible. And I'm really grateful to the amazing speakers that we have joining us today. I want to recognize especially Cami Martin and Misty Good, who both serve on the NEC Society's Scientific Advisory Council and are really critical to the work that we do. And I also want to highlight Nick Embleton's work because he has led a project called the Butterfly Project that really recognizes the trauma of bereavement and neonatal loss, and especially what it's like to have one twin survive and one child pass away after a NICU experience. And so again, if you'd like to learn about Nick's work, you can do that by checking out that link in the chat box. So I'm going to pass it over to Cynthia, and just want to quickly remind everyone that we are not providing medical advice and that all faculty views are independent from the NEC Society. So Cynthia, I'm going to pass it over to you, and thank you so much again for being here, everyone.
Anyway, hi, I'm Cynthia Bear. I'm the editor-in-chief of Pediatric Research, and I'm delighted to be co-moderating this session for the NEC Society. And just a few words about Pediatric Research. We're the official journal of the American Pediatric Society, the European Society for Pediatric Research, and the Society for Pediatric Research. We publish the full spectrum of research that touches children's health. So anywhere from basic to clinical to epidemiology, health services research, epidemiology, QI is part of the research that we cover. We also have a very robust, what's called the front half of the journal, which are editorials, commentaries, policy commentaries, letters to the editor, which we call correspondence, family reflections on how this research touches families and impacts them, and images of mechanistic pathways that might be useful for people in their presentations. Our cover art, which I'm showing in this slide, is chosen from the original research articles. So if you're one of those people that has always wanted, a picture of your research work on the cover of a journal, here's your opportunity to submit it to pediatric research and get it on the cover. We have very quick turnarounds for the industry. Time to first decision is less than 40 days. Our impact factor from 2020 was 3.75, and we have the full resources of the Springer Nature Publishing Group at our fingertips to help with getting your article out in news press and press releases and widely advertised. So there's our website if you want to visit us. In the world of publishing, most journals now are going to online only editions. So you'll find all our recent articles there, the cover images and other resources. So thanks for inviting me, Cami, to be part of this. And I'm going to turn it over to you now.
Thank you so much, Jen and Cynthia. And so welcome, everybody. I'm Cami Martin. I'll be co-moderating this session with Cynthia Bear. And thank you for joining us today. So this is going to be the first of 3 webinars, and we'll share the dates of the second and third one near the end of the session. And so each speaker has a challenge of telling us all about their expertise and everything they know about NEC in about 15 minutes, with leaving some questions at the end of this 20-minute block. And so I remind you, for any questions, please use the Q&A box so it doesn't get lost in the chat. And we'll be moderating the questions and pose them to the speakers at the end of their talk. Also, just so you know, if you're looking for general background slides of necrotizing enterocolitis, I did ask the speakers to jump right in. We're all very familiar with necrotizing enterocolitis and really wanted them to highlight their research, discuss the hot topics within what they're looking at. How can we learn from these findings and move forward in advancing research, as well as advancing care for those at risk for necrotizing enterocolitis? So very excited to hear all of them. Thank you also to all of the speakers, and let's begin. So our first speaker is going to be Dr. Venkatesh Sampath. He is a professor of pediatrics and medical director of the Donald Thibault Lung and Immunology Laboratories at Children's Mercy Hospital. He's an accomplished physician scientist studying necrotizing enterocolitis and bronchopulmonary disease with an expertise in innate immune signaling. And so today he's going to talk about genetic susceptibility and NEC.
I want to thank the NEC Society and Pediatric Research for this great seminar they've arranged. And also thanks to Cami for this invitation to present some of our work. I have no conflicts to disclose. In this very brief talk, what I'm going to try to do is, rather than focusing on the whole genetic base of NEC susceptibility, I'm just going to walk you through some of the data we have on how genetic mutations can program abnormal toll-like receptor signaling in necrotizing enterocolitis, and essentially use the knowledge we've gained from studying a single gene called SIGR, or single immunoglobulin interleukin-related receptor. I would also show a lot of our unpublished data, which essentially suggests that SIGR regulates postnatal intestinal adaptation. It's very important for mucosal barrier establishment and gut microbiota composition. And the data we present will be from a special mice we made, where we knocked in a human SIGR mutation that we found from an infant who passed away with NEC. And then hopefully at the end, I can lead you to believe that if you have severe genetic defects in the inhibition of toll-like receptor signaling in the gut, and that can program NEC susceptibility. Essentially, if it's SIGAR and genes like SIGAR act as a brake against NEC in the intestine, and if you have mutations in those brake genes, you can get NEC. I took Cami's suggestion and jumped right in without much of a NEC background. So just to get people caught up, I think what we think happens in NEC, or what are some of the common theories for NEC, is that bacteria in the intestine in preterm babies can activate toll-like receptor 4 signaling, and that activates inflammatory cascade, resulting in severe inflammation, intestinal NEC, and injury. Now, while this is recently established, the interesting part is that things like intestinal dysbiosis or abnormal microbiota and intestinal injury are shared among a lot of preterm babies. So the question really is, why do only some infants get this very abnormal inflammatory response and response to bacteria in the gut called NEC? And importantly also, why do some kids get very severe disease? And really right now, we don't have any ways of understanding these things. So the part which is less appreciated is that there are a lot of breaks in the system in adult intestine and also in the neonatal intestine. Like there are multiple genes like SIGAR, A20, multiple microRNAs, which are known to repress TLR signaling. So one of the hypotheses that we came up with is that perhaps in those kids who get NEC, there's a problem with these breaks and resulting from genetic mutations. And if you have a problem with these genes which regulate TLR signaling or suppress TLR signaling in the gut, those are the kids who actually have a propensity to develop NEC. So our hypothesis is that it's a failure in the breaks. So I'll start with a clinical scenario, because this is really what, like the NEC Society, inspired my work into SIGAR about 7-8 years ago. I was looking after a twins of 23 weeks, and one of the twins at day 52 of life developed very severe NEC and passed away with NEC totalis. His twin also developed NEC 7 days or 8 days later, but had just severe stage 2 NEC, never required surgery, but survived. I took a chance, and what we did was a forward genetics approach, in which, what does that mean is that we took some babies who got NEC. We used an unbiased exome sequencing approach to identify whether some of those kids might have a genetic mutation in some of these TLR genes or genes that inhibit TLR signaling. And we are fortuitous that among the 3 or 4 kids we sequenced, one of these twins had a mutation in a single immunoglobulin interleukin-related receptor, or SIGAR. And it was fascinating to me because, I was developing this hypothesis that mutations in these brake genes, SIGAR, A20, would potentially contribute to disease, and SIGAR is essentially that. It was described in 2002 by Georgia Lee's group in Cleveland Clinic, and she showed that SIGAR is a major regulator of TLR signaling, inhibitor of TLR signaling in the gut. So this gave us, suggested that our hypothesis was correct. Here, what I show is the SIGAR structure, and this becomes important to understanding how SIGAR works. A lot of these on the right, the bigger figure maps all the domains of SIGAR, the extracellular IgG domain, the transmembrane domain, and the intracellular domain. All these small lettering and maps all the mutations that have been already described in the Human Genome Projects in SIGAR. And for us, today we'll talk about this one mutation, the SIGAR1 stop mutation we found. This stop mutation is a large, because it's a stop mutation, it really disrupts the SIGAR protein, that most of the intracellular domain, which is very important for inhibition of toll-like receptor signaling, is essentially lost. So what we did is that we initially developed a very simple assay to check for the functional nature of this mutation. Finding variants or mutations in the human genome is very common. All of us will have more than 2 million variants just at the exonic level, but most of those mutations don't cause any harm. So it's important to understand how mutations affect function of the protein. So the assay we developed was very simple. We would we would treat cells with LPS. LPS is derived from gram-negative bacteria, which are hugely implicated in NEC. And then we have tools that we can put any protein we want, we can express it in the system. So in this case, we put the wild type protein, SIGAR protein, and the stop mutation. And we could look at inflammation and cell death and that kind of thing. And the first thing that we realized is that, here, the first picture on the top is NF-κB activation. NF-κB is very important for the pro-inflammatory response downstream of toll-like receptor signaling. And you can see that when you add LPS to the system, NF-κB activation goes up. When you put the wild type, the normal cigar protein, it represses inflammation by about 50%. And if you put the stop mutation, you have complete loss of function of the cigar protein. So the inflammation is actually back to baseline. In some cases, we have noticed that it even interferes with normal cigar and makes the inflammation even worse than baseline. And you can look at IL-8 activity, and IL-8 activity also shows that LPS induces IL-8 in the cell line. And then you put the wild type protein, cytokine expression is decreased, inflammatory cytokine expression, and this is lost with the mutant protein made by the mutation we found. And here we're looking at CC3 or cleaved caspase 3, which is a marker for apoptosis. Remember, LPS or toll-like receptor signaling, when it's excessive, can cause cell death in the intestinal mucosa, and necrosis is a big part of NEC. And LPS induced CC3, which is a marker for apoptosis or cell death. The wild type protein suppresses this by more than 60–70%, and the mutation actually causes back to increased CC3. This is all a Western blot analysis. So this led us to conclude this mutation we identified was a real bad mutation. It results in complete loss of function in cigar, or at least in cell lines, causes exaggerated TLR4 inflammation, or at least in a minimum does not suppress TLR signaling. So what I've shown you now is a bedside to bench approach where we looked at what is called a forward genetic approaches, unbiased approach, and we went from the genome of the child who got NEC and identified cigar as a potential locus for NEC. But however, we want to understand how do cigar mutations program deviant host microbiome interactions in NEC in a live model. So we used a mouse model, which is a commonly used model to study NEC pathogenesis. But before jumping in, what we wanted to show is that how is cigar expressed in the mouse intestine? On the left, the immunofluorescence image shows that there's hardly any cigar expressed on day of life 4 in the mouse intestine. At 7, it starts expressing itself. At day 14, it's very nicely expressed. And you can see the green, all the green color is on the brush border mucosa, intestinal epithelial cells. So it clearly shows that cigar has a temporal expression pattern in mouse. And this is very similar in humans. This is a human tissue at 34 weeks from a baby who died from other diseases. And all the brown stain you see shows a cigar expression. And this is important, as we know that NEC is more common in infants less than 32 weeks. And in mouse, it's very difficult to induce NEC after a certain stage because they start expressing more, their expression of several genes mimics the neonatal gut or term neonatal gut. So we identified cigar as like temporally regulated, and its expression is low in the window where NEC can develop, both in humans and in babies. So then we actually took a leap of faith, and we want to understand how this mutation causes NEC. So, using CRISPR-Cas9, we worked with Aaron Gert's lab in MCW, and we knocked in this mutation using CRISPR. And the most important blot here is to look at is the blue blot on the right extreme. And you can see the normal cigar protein is expressed at 55 kilodaltons. And if you're homozygous for this mutation, you make the truncated protein, which runs at 18 kDa. Now, these kind of mouse models are powerful because this expresses exactly the mutation and allows us to study what exactly happens in the intestine with this mutation. It's close, almost the closest you can get to what we can study in humans itself. So we came up with the hypothesis that maybe, how does the cigar mutation cause NEC vulnerability? Now, it could impair the mucosal barrier. Now, this simplified cartoon is not fully accurate because normally bacteria don't come in this close to the intestinal mucosa. There's a layer of thick and then thin mucus. But eventually, when this mucus barrier is disrupted, the bacteria actually come in close contact with the intestinal epithelial cells and can activate inflammation and cell death. So cigar mutation could impair this mucosal barrier. It could also, when humans are born or mice are born, there's a big adaptation going on because the gut is not really, in preterm babies, it's not really meant to be exposed to so much bacteria. So there's a lot of mechanistic changes happen to allow us to live with bacteria in the gut. And so cigar could affect that too. It could also cause exaggerated TLR-mediated inflammation or sensitivity to bacteria in the gut. It could cause aberrant gut colonization. So really, we went in with a multi-pronged hypothesis as to how cigar mutations can cause abnormal next susceptibility. So the first experiment we did, and this is a really cool experiment which got us all excited, is that we didn't do any experiments. We just took day of life 8 mice, and we took littermate mice, which means all the mice are from the same litter. We can cross mice in such a way that you can get all 3 genotypes in the same litter, and that's a very good control because they're in the same cage, same mother and father. So it's as good as control you can get. And you can see that even without experimentation, compared to the wild type mice, which are in green bars, if you lose 1 copy of cigar and it's replaced by this mutation, you get increased IL-1 beta, TNF, and interferon beta. By the time you lose both the copies of cigar, you actually see a 3-fold increase in a lot of these cytokine genes. And note that IL-1 beta is a very prominent cytokine, and even in human studies, it goes up very highly in NEC. So this got us excited. So we actually did a Western blot analysis where we took the ileal tissue, ground it down, and it's a way of looking at protein expression. And the top red bars here is PP65. That's NF-kappa B. NF-kappa B is a transcription factor that's activated with inflammation, and phosphorylation is a sign of activity. And you can see even at baseline, without any injury in the same mouse family, if you have mutations in cigar, there is actually more expression of this phosphorylated protein, suggests this low-grade activation of NF-kappa B, which would be consistent with the inflammation we're seeing. So we thought, okay, maybe this low-grade inflammation could happen from 3 reasons. One is there could be a defective mucosal barrier, allowing bacteria to come very close to the intestinal mucosa and triggering the inflammatory response. Because of dysbiosis, and normally we think that it is the feeding, the different factors which cause the shift in the microbiome, but it could be the host genetics plays a role in that too. So could cigar mutation cause a dysbiotic picture, and could it impair postnatal intestinal adaptation? So we looked at the first question first, which is how does postnatal gut adaptation impaired with the cigar mutation. All this work was done by Wei Yu, a talented PhD in my lab. Essentially, in very simple terms, when bacteria activate TLR4, they have to go through multiple mediators before they can activate inflammation. And what the body has figured very smartly to do is that this gene called IRAC, or this protein called IRAC, is very important for this inflammatory cascade. Now, if the body has actually figured out a way to suppress the expression of IRAC after birth. So when IRAC is suppressed, this whole link in this chain is disrupted, so you don't get inflammation. So the question then is, does cigar regulate IRAC? And we just proved this in a nice paper. And here we have day of life 11 ileal sections. And you can see in cigar, the wild type mice or normal mice, you get some expression of IRAC. If you lose 1 copy of cigar is replaced by the cigar mutation, which I've been showing you data on, you get some IRAC expression. If you lose both these copies, there's a lot of IRAC. Now, so essentially this postnatal adaptation where IRAC is supposed to go down doesn't really happen when you have the cigar mutation. And subsequently, we've done some mechanistic studies to show that cigar regulates STAT3 and microRNAs, and that is the cause for this IRAC suppression. But the key finding here is that cigar is regulating postnatal adaptation through IRAC. So the next question we had is that, is there a defective mucosal barrier in cigar transgenic mice? And one way of looking at this is looking at the expression of intestinal adhesion molecules or tight junction proteins. When cells, whether it's lung cells or gut cells, these are kept very close in close proximity to each other because of the expression of adhesion molecules. And when these molecules are expressed in less, what happens is that the cell-to-cell contact is sort of becomes leaky and bacteria can get across. So this is work done by Aparna Venkataraman in my lab. And essentially what you can see is that compared to the wild type mice, the homozygous knockout mice, whether it's ecadrin, epchem, or claudin, which is a tight junction protein, all these proteins, which are normally expressed and are very important for maintaining the mucosal barrier, have less expression in the cigar mice, suggesting that they'll be vulnerable to to bacterial translocation. Now, we're doing experiments to prove it, but at least these data suggest that this will happen. We can also go into the colon, and you can see mucin, which is a major component of the mucosal barrier. Expression of mucin is also decreased. So this suggests that cigar regulates the expression of cell junction proteins and mucus, which are important for maintaining the mucosal barrier. So last, we really want to explore this hypothesis. I've had this for many, many years, that how does cigar directly or host genetics directly regulate the developing microbiome? Because that would be a cool finding. So it's not just the formula feeding, other things that we do externally. It could be the host genetics itself regulating the host microbiome. So we did 16S sequencing at day of life 14 and 28 in mice. And the first things I wanted to show you, which is interesting, is that normally all our human studies, we would focus on the stool from babies, and that's probably more reflective of the colonic microbiome. The ileal microbiome is actually quite different. Though there's some overlap, I'm sure that's coming from the terminal ileum, the ileal microbiome is quite different. That's not what this figure shows. And also, the day 28 microbiome is very different from day 14. And the way to look at this is that after a baby is born, the initial microbiome at 4 weeks is not going to be the same as the microbiome in the same child at 3 months of age. So that's also been shown. And this has been shown by multiple groups, including Washu and other researchers. But we then went to the most interesting question for us, and that is, does the cigar genotype itself regulate the microbiome? And again, these are untreated mice. We didn't do any experiments with them. We just took their colonic microbiome, did 16X sequencing. All of them are from the same litter also, or multiple litters. And you can see, even with this, just the change of the genotype, we can see differences and we had good numbers, about 7 to 8 per group, and we could choose some changes in the colonic microbiome with a p-value which is significant. And then also in the ileal microbiome, we can also show that there is a significant change based on the genotype. And for this comparison, we compared all the wild type mice with mice which either had 1 copy of the mutation, 2 copies of the mutation. This mutation is pretty powerful, that even 1 copy of the mutation tends to have pretty severe disease or moderate disease. So we put the groups wild type versus no mutation together, and we can see this. So next, then we induce NEC in these mice, and this is a common model people use. They take these mice and give them formula feeding, hypoxia, and endotoxin enterally, and this causes intestinal injury. And interesting, the first table there shows that in this experiment, and we are looking to get at microbiome changes. The microbiome changes in this model is a combination of the experiment, whether you got NEC or you didn't get NEC, whether you had a genotype, a cigar mutation or not, and the tissue type, whether you are we collect the microbiome from the colon or the ileum. And you can see the experiment itself changes the microbiome as expected. The tissue type or the colon versus ileum also changes the microbiome. But despite these huge influences, the genotype also influences the microbiome. And interestingly, when we compare the cigar homo mice with the heterozygous mice, there is no difference in the microbiome, despite the NEC and the tissue type. But when you compare homo mice with wild type mice with the heterozygous mice, 1 copy in wild type mice with the wild type mice, these are all significantly different. And we are doing further studies to analyze this. And I bring this, so it's a
One more minute, Dr. Sampath.
How many minutes, sorry?
1 more minute.
Okay, so I'll jump to the conclusion then. We showed that there's more enterobacteria in basically cigar knockout mice. And then NEC vulnerability also is induced, is depending on the mice. So if you have wild type mice, you have some NEC. If you have both mutation, you have more severe NEC. So essentially what I want to show is that cigar variants, cigar facilitates neonatal adaptation. We have pilot data showing that cigar mutation disrupts cell adhesion molecules that maintain gut barrier function. Cigar mutant mice have mild dysbiosis at baseline that is exaggerated after experimental NEC. we speculate that severe genetic defects in TLR inhibitors like a cigar can program deviant host microbiome interaction. So we think cigar is one of the genes which puts a brake against NEC. If you have mutations in those genes, you are vulnerable to NEC. I'll stop there and thank my wonderful team for all the work they do, which allows me to be come across as being smart here. And if there's time, I can take some questions.
Yeah, no, that was absolutely fantastic. And thank you so much. Yes, let's open 2 questions. Do we have any yet in the chat box? I have
Yes,
one or two, but I'll wait
do.
for you.
We have a question in the chat box. So that was a great talk. Thank you so much. We have a question from Juan Matute. Do you have details of the ASV OTU species that drive the difference between the different genotypes of cigar?
Yes, we have data. I actually have a slide on this, but I can tell you that one of the data I rushed through is that Enterobacteria seems to be prominent in the cigar mutant mice, with 20% of the bacteria in the cigar clone mice is Enterobacteria, whether it's only 2-3%, and that seems to go higher with NEC. So we do have it. Because we are short of time, I didn't show the data. Yes, but we are working on the further analysis too. Thanks. Nice question.
All right, I have a question for you, and then I think Cami has a couple of questions. So my question is, I thought it was very interesting at the beginning of the talk that twins, one of whom got NEC and the other of whom did not, were discussed. And then you're talking about a genetic susceptibility. And my question is, has anybody ever done twin studies looking at paternal versus identical twins to actually see if there is a genetic susceptibility?
Yeah, actually, Vineet Bhandari was the first one who showed that in a series of studies in 2004, 2006. And they did twin studies, and they suggested that, about 50% of the variability in NEC susceptibility is genetics. And they did exactly what you're suggesting, which is they compared non-identical twins with identical twins. There's also case reports which frequently come that both twins got NEC within the same period. So, and we see that sometimes too. So there is clearly twin studies showing that there is some increased vulnerability of identical twins compared to non-identical twins, whether it's fully genetic or shared or component of shared environmental factors. That's not been deleted, but there's enough. There's some papers on that too. Yeah.
Thank you. Cami, do you want to ask your questions?
Yeah, and I'll just stick it to the one question, and then we can move on. But really enjoyed your talk, and as within nutrition, I think I'm increasingly getting more and more appreciating the effect of genetics on metabolism, and this is really a great example. The effects of that and mutations across so many domains. So the question is as all of us investigators are trying to do this comprehensive characterization of the disease, especially the question of, different pathophysiologies, different definitions may not be one disease. Do you suggest in future investigations that, especially omic investigations, that this type of genetic analysis and looking at genes and polymorphisms are part of that panel to really be able to make sense of the data?
I do think so. I think it's going to take a lot of work. As you know, Cami, I followed some of your work too. Getting one part of the study done it's also so difficult. And then people are studying so many groups, but really the study is going to be, the most definitive studies are going to be studying the microbiome, the metabolome, and the genetics as one big factor. Now, it's going to take 3,000 babies to do it. And one of the things that I've liked about the NEC Society is that they're bringing really a lot of groups together. So in times that we are able to do it. But I agree with you. So there's probably at least 2-3 different types of NEC we see even in preterm babies. And that might come from an ischemic insult or mucosal insult or genetic-based susceptibility. And categorizing, actually, and we have data in case, I think we should talk offline too. We have data that some of these compounds, metabolites and long-chain fatty acids, has altered in these cigar mice. So it's
interesting.
It's question is which comes first, but maybe that's not important. It's probably happening at the same time. And but clearly host genetics is changing the microbiome and the metabolite component too. So we have data that lactate and long chain dextrohexanoic acid is altered in these mice. So I agree with you totally, but it's going to take some significant will and effort to do all those, to put those 3 things together to answer the questions. Yeah.
Yeah, no, that's great. You'll definitely hear from me. I'd love to talk about that. And this is exactly what we hope to accomplish through the Next Society is build this community so that we start to collaborate together and get those numbers and interest and moving forward. Terrific.
Cami,
Okay.
Cami, we have several more questions that
Oh.
got posted a little late. Do we have time to?
Well, what we
continue?
can do is, I think, Venkatesh, I think you can see the chat and he can answer them offline.
Actually,
Or,
they're
he
in
can
the
answer them
Q&A
now.
now. They're in the
Yeah.
Q&A.
Okay,
You can see them?
I'll go,
Great.
I'll go through
Now-
those and do that. Yeah, thank you.
Okay, thank you so much. All right, Cynthia, go ahead with the second speaker. Thank you.
Okay. I am delighted to introduce Misty Good today as our second speaker. Her talk is modeling neck gut on a chip, which sounds very interesting. Recently, Misty moved to the University of North Carolina, and congratulations, she's the new division chief of neonatology there. She's also a member of the Scientific Advisory Council of the NEC Society. And one credit to her, a very large credit, is that she has established the multi-site neck tissue biorepository. So Misty, take it away.
Thank you so much, Cynthia, for that kind introduction. While I'm pulling up my slides here, I want to thank the NEC Society and also, Peds Research for having me. Can you guys see my slides okay? Yep.
Perfect.
Oh, perfect. Okay, so I'll be talking about gut on a chip and how we can model that in the laboratory using patient samples. And so just first, my funding disclosures. We've been really fortunate to have a lot of great funding from the NIH, NEC Society, and others. And certainly institutional support over the years has been wonderful to us. So thank you for that. Brief outline of the talk. So I'll introduce you to what are these organ on a chips that we'll be talking about. And they're really these small microphysiologic systems that are really quite powerful and really the next stage after organoids. And I'll talk about a brief overview of our in vitro preclinical neck models that our lab and others utilize, and then the limitations of those, and that is what created or inspired our creation of the neonatal intestine on a chip model and also the neck on a chip model, and then also how we can utilize these intestine chips as a personalized medicine approach. And so just to give you just some up close pictures of what exactly this is. So they're all patient derived. So we collect pieces of intestine from the operating room when babies have surgical resection for neck or other indications. And what we do is grow them up in the lab over several days, and then they go onto this chip that you can see on the left side here. Now, these chips, if you look in the upper right-hand corner, are broken up into two different chambers. So there's a top epithelial cell chamber and a bottom endothelial cell chamber, and there's crosstalk in the middle via porous membrane. These chips go into their chip house, which is a pod, which houses all of their media, nutrients, drugs, if you want to do drug discovery, etc. And then these pods then go into this Zoe culture module here. And what we can do then is regulate the flow of media into the chips, which just like in our own intestines, mimics the intestinal flow. And then also this orb module on the right here sends signals to the Zoe, and that actually stretches the chip similar to peristalsis in our own intestine. And how does this look like under a microscope? So this work really was originated by Dawn Ingber's group out of Boston. And so these are some really elegant studies that they performed where they took adult human duodenal organoids, grew them up into a lab. So organoids are these little mini guts you can see here, co-cultured them in the chip with these human microvascular endothelial cells. And again, this is what the chip looks like here. And it's you can see in cross section, the top chamber, here's this epithelial chamber, endothelial chamber on the bottom. This is and C is a scanning electron microscope, so you can see just how these little villi are actually forming. And then down here you can see in magenta here, this is zona occludens 1, which is one of the tight junctions. So this is just showing the epithelial cell chamber and how all the cells are confluent there. And then cadherin shown in green is the endothelial cell chamber. this isn't necessarily important, just want to give you a brief overview of that. And then what they were able to do is, because there's 2 chambers in the chip, you can actually measure how something that you place into the top chamber flows into the bottom chamber, and is that different between different patient donors, which is really exciting, and leads us to a personalized medicine approach. And so how do these intestine chips, develop from these little mini guts that we have in the lab? So this is some additional work from that group showing over certain time points. So at 4 days, with the addition of flow here, you see nice confluent cells here. By 6 days, you start to get these little invaginations here. By 12 days, you're really getting some deep crypt-looking crypt-like structures, but without flow, you're really stuck at 4 days here. And so that's really just similar to a typical cell culture model in the laboratory. And what this group also showed is that there's certainly multiple cell types that are present in the gut on a chip. And so, can we really recapitulate the human situation on this chip? They wanted to know, do you see all the different cell types? So including enterocytes or intestinal epithelial cells, goblet cells, enteroendocrine cells, Panus cells, and stem cells, all of which are impacted and important in NEC. And they definitely saw that. And so we all know why NEC happens, as Cami mentioned, but I wanted to put this slide here just so you can see, how do we go from an adult intestine chip to then a premature intestine chip, and how can we model in the laboratory based on what we know about NEC pathogenesis already. And so we all know that the microbiome is really important, thanks to Barbara Warner and Phil Tarr's work and others in the field that have shown that really 48 to 72 hours prior to a diagnosis of NEC, our babies will get a proliferation of pathogenic bacteria or dysbiosis of the gut. And so we wanted to be able to mimic that in the laboratory on this chip. And so, we do culture the intestinal bacteria from the resected pieces of intestine that we get in the laboratory. And basically what happens at the cellular level is when you co-culture intestinal epithelium with this dysbiotic microbiome, you get breakdown in these tight junctions that are important for gut barrier function. So you get a leaky gut, that bacteria gets across the gut barrier, what we call bacterial translocation, and really exaggerates the pro-inflammatory response that we all see in human NEC. And so in vitro NEC models have certainly come a long way. So we certainly start with premature or term infant intestine, because we know that there are term babies also affected by NEC. And what we do is then isolate the intestinal stem cells out of the crypt. We culture them up in a dish with various growth factors, and then we can subject them to that dysbiotic microbiome, so what we call NEC in a dish. And I'll show you why that's a great model, but there are some limitations. These are just what some enteroids or intestinal organoids look like under a microscope. Some nice pictures there. And why is this important? So we can take these little mini guts or enteroids and we can feed them, okay, with breast milk or donor breast milk shown in this slide here. And so they can uptake nutrients, and also, you can co-culture with personalized microbiome. And so we all know that ways to enhance mucosal healing and intestinal stem cell regeneration, such as with breast milk, we know breast milk is protective. Several investigators have now looked at all the different ingredients and shown why, but we know that this can either attenuate or prevent NEC in humans. And so one of the things that over the last couple years that has been important in enteroid work is how the enteroids are cultured and in what configuration. And so really this group, Co et al, and Cell Reports, that I encourage you to look if you do any enteroid work, published a really nice, elegant paper in which they show that all the enteroids that we have been culturing up in the lab are really basal out, meaning that this is the basal side of the epithelial cell. And they were really one of the first groups to show that you could do some things in the lab and get an apical out configuration. And why that's important is because when you are co-culturing with microbiome or trying to infect the cells with virus, for example, or bacteria, you really want to make sure that the nutrients are actually getting taken up. And so, so for our NEC in a dish model, then what we originally were doing was culturing up those basal aneroids and subjecting them to what we call nectaria, which is the premature infant dysbiotic microbiome that's known to have a preponderance of proteobacteria. I'm happy to talk more about that later, but just for lack of time, you can see that when we co-culture these aneroids with the nectaria at 48 hours, you see a breakdown of E-cadherin, or one of the tight junctions that basically holds the intestine together. So this is the neck on a dish model. And one of the things is that when we first started culturing with these particular basal aneroids, then we cultured apical outs. The apical outs, and I'm not showing the data here just for lack of time, but the apical outs died within by 10 hours. So certainly very different. And why that's one of the limitations is, you can only do so much. We know that our babies, some of them get very, very sick and need surgery within 8 hours, but some of them do not. And so being able to model this appropriately over several days in the laboratory so we can see the evolution of what's actually happening at the intestinal level is really important. And so we sought to create this neonatal intestine on a chip at first. And so we cultured our premature aneroids like we do, you can see here, and this is work done by a very talented now medical student who was a technician in my lab, Wyatt Lonick. And what he did is take these aneroids and then dissociate them onto the gut on a chip, or onto the chip, to make a gut. And you see after 3 days, there's this neonatal epithelial monolayer that then by 6 days, you start to get those deep invaginations here. And by 8 days, actually, you can see the villus-like axis, which is, fantastic. And then just to give a schematic here of this, so our regular neonatal intestine on a chip that you just saw, we add that enterobacterial slurry, or that dysbiotic microbiome that I talked about further, and then look at different readouts, including pro-inflammatory cytokines, reduced epithelial proliferation, and gut barrier integrity that are really important in the human disease. And so first we wanted to see, just like I had mentioned when the adult gut on a chip was first, presented, we wanted to see, are all the epithelial cell types present that we see in human intestine, in human premature intestine, but also during NEC. And so in the first column here in red, you can see staining for goblet cells, the second column Panna cells, the third column endocrine cells, and the fourth column Ki67, which is a marker of proliferation, all shown in red. And you see in human controlled premature intestine, you can see that those are their nice cell types there, but in human NEC, all those cell types are certainly have decreased expression. And then in neck on a chip, you also see in our control chips, you see nice expression of these different cell types. And then in our neck on a chip, you see that those are decreased, suggesting that the neck on a chip can recapitulate the human disease. And then we looked at a gene expression level. We looked at epithelial cell markers, including stem cell markers, but also some of the markers we looked at in the previous slide, but also pro-inflammatory cytokines such as IL-1 beta and IL-8, which Venkatesh Sampath had mentioned, are very important in neck pathogenesis, and also NRSI proliferation, Ki67 and PCNA. And when we looked at our neck on a chip, we see that they're very similar gene expression, and this gene expression in the neck on a chip actually mimics the human situation. And then we really wanted to take an unbiased approach. So it's fine to look for, a few genes here and there and do they match up, but we really wanted to take an unbiased approach to this. So we performed what's called RNA sequencing on our epithelial cells seen in the chip at different time points. And we wanted to see what are the different pathways that are upregulated or downregulated in this neck on a chip model. And importantly, several of us know that there's different, all different cell types that are present in human neck, including apoptosis, necrosis, or excuse me, necroptosis, also necrosis, and ferroptosis. We also published a paper this week with collaborators of ours on a new cell death pathway that's seen in neck called lysoptosis. So I encourage you to check that out. And one of the things that we're really interested in the lab is cytokine signaling and the cytokine receptor interaction, which is upregulated, but including the pro-inflammatory pathways, TNF signaling and IL-17 as well. And this is just a really zoomed out approach showing that there's over 5,000 genes that are altered in our neck on a chip model. And then we are working on comparing those to the human situation as well. And then we wanted to look specifically at the pro-inflammatory gene expression. And you can see that at 24 and 72 hours, shown in red here, there's several pro-inflammatory genes that are upregulated. We also looked again at just talking about cell death, since it's so important in this disease, and we see significant upregulation of the cell death pathways as well. And then, as I mentioned before, calculating the gut permeability of the chip is really important. So we can put a radio label tracer at the top of the chip and then see over time when that tracer gets into the bottom chamber, okay? And so that measures the permeability. And so when we administer the Nectaria or the dysbiotic microbiome to the chip, you see over time gut barrier function is really impaired.
Misty, this is your 1 minute warning.
Okay, thank you. So how this looks pictorially, you can see here, ZO1, these nice tight junctions, and then those are impaired in the chip at different time points. Things that we're working on now is looking at personalized responses to NEC on a chip and how different babies will actually respond to this dysbiotic microbiome, looking at variations in their pro-inflammatory response. And we're excited about this model as a promising new patient-derived model of NEC. It recapitulates several features of the disease, and we are using it now as a drug discovery platform as well. And I think we accomplished our objectives. And this is my plug to Fight Like a Preemie for all of us researchers who are doing this basic science research. And we're really working hard in our lab to, as the NEC Society says, build a world without NEC. So I'm happy to take any questions, and if you're interested in joining my lab, please reach out and email me. Thank you so much for the opportunity.
Terrific. Thank you so much, Misty. We do have a question here. It says, thank you for your great talk. Recent gene expression data on intestinal organoids suggests that TLR4 may not be expressed in ileum organoids. What is your thought on this, and have you measured TLR4 in your model?
That's a really great question. There's a lot of fantastic work that's being done with David Hackham's lab and others, including Venkatesh Sampath, on toll-like receptor 4 specifically. So we have measured TLR4 gene expression throughout our biobank, and actually the data is really variable as far as human gene expression. For the most part, though, in human NEC, and we all know human NEC is so variable because it depends on when the surgeons decide to operate on our babies. And so that can really affect the gene expression as well. The timing of upregulation of TLR4 is definitely very important. So we certainly see it in upregulated in humans, not every human. And just as I briefly mentioned on that slide, you can see that there's significant variation in just how those were actually babies with NEC, how their pro-inflammatory responses, some have gene expression way at the top, and the others have a bump compared to baseline. But really, it's quite variable, which is why our science is, imperfect, but we're trying.
That sounds good. We have another question actually from one of our speakers for a future webinar from Christopher Stewart. So he passes along your congratulations. So for organoids in the experiments, did you differentiate the organoids before experiments? And then a topical question that we've been hearing a lot about and the influence of the microbiome, and that's the oxygen gradient. So do you know the oxygen gradient in these co-culture models, especially when adding viable bacteria? Is the oxygen gradient across the epithelial recapitulated in your model?
That's a great question. So one of the issues with chips in general is that they're very permeable. So depending on there's a bunch of different chips, but depending on the ingredients that make up the chip can really affect that oxygen gradient. So these chips actually, I didn't show the data, but we have also subjected them to various ranges of hypoxia. And so, there's suggestions, right, that babies that have desaturations all the time, and certainly in our mouse model, it's one of the things that's required to produce mouse experimental NEC is, having this hypoxia. So the long-winded answer to that is we haven't measured it, the gradient across the chip, just because our chips are, they take up whatever their environment is around. So if they're at room air, they're going to be 21%. If you're culturing them and 1% oxygen, 5% oxygen, that's going to definitely affect cell viability over time, et cetera. But I didn't show the data, but they can, these chips can survive in a hypoxic environment. And certainly when you're looking at different probiotics or microbiome, challenges to these chips, that's going to be really important.
Another way, they're coming in fast. So let's do a couple of quick ones and then we can move on. And Misty, go ahead and answer them during the next talk. Absolutely. But let's go to the next one. Is there a robust mucus layer developed on your chip model?
That's a great question. There's definitely mucus that's present. There are different drugs that can stimulate or hinder mucus development. And so, we can exaggerate the mucus development and then look at how the colonization of the different microbiome that we can add to the chip in particular, how that plays into it and how that can affect. Since I just moved recently in the last 2 months, those are things we're working on currently. So thank you.
Yeah, and this might be related to this last question. So sorry we couldn't get to the others, but Misty will answer those. Is there a time window where you can study reversibility? By example, what you mentioned, probiotics in your model.
That's great. We're actually working on those studies currently. Like, at what time point can we either give a drug or give a probiotic to the chip and reverse the disease? I will say the model is very severe, and so we're working on different variations of the model to not make it so severe. But when we first started out, we wanted to make sure that it was something that was similar to our human disease. And now it's so severe, we're scaling back to see if there's different time points in which we can intervene.
Perfect. Well, thank you so much, Misty.
Thank you so much.
All right.
Appreciate it.
So let's move on to our third talk of the morning. In this, I would like to introduce Dr. Agostino Pierro. He's a professor and head of the Division of General and Thoracic Surgery at the Hospital for Sick Children. A rich, long-standing, accomplished work, especially research directed towards neonatal intestinal disorders. And he's going to present today some recent work and some thoughts on novel therapeutic strategies, specifically remote ischemic conditioning. Dr. Pierro, please begin.
Thank you very much, Cami, for the nice introduction. Can you see my slide?
I can.
Okay, well, thank you much, and sorry for my voice today. So I'm going to show you something that we in our lab, we've been quite excited about it, and possibly a new way of treating NEC in the future, remote ischemic conditioning. Sorry. Okay, I have no disclosure to make. This is the challenge, and we all know about it. And if you look at our mortality rate in Canada with the CNN, the Canadian Neonatal Network, we could see that the mortality for surgical disease or medical NEC or the overall mortality is unchanged over the years. So it is, in my opinion, and the opinion of others, that treatment and outcome has been unchanged. We need a novel therapy to try to influence what's going on in these babies. So how can we avoid, I'm a surgeon, so I'm going to show you some dreadful picture here, but how can we avoid to go from the left panel where we have a perforated neck to a more patchy areas of necrosis in the bowel to a very panintestinal disease? Can we avoid that? And this is what our lab has been working. We've been fortunate to document this in a paper on remote ischemic conditioning in the way of counteracting what's going on in NEC. So what we found initially in human babies is that if you look at area most affected by the disease compared to the one farther away, where we actually sometimes perform the anastomosis or the ileostomy, you could see that there are macrovascular impairment in both hypoxia and macrovascular impairment in general in this baby. So our question was, what can we do to prevent this phenomenon? And that's why we took our attention in remote ischemic conditioning, which has been actually described for reducing the cardiac damage. There have been also clinical trials to demonstrate the benefit of that. But nobody looked into the intestine as such. So what is remote ischemic conditioning is very similar in a sense to measuring blood pressure. So an extension of is a very safe and feasible probably way of treating patients by doing this maneuver. So our hypothesis was, can we actually do this and see if we could protect the intestine from further neck damage? And this is the hypothesis we brought forward for all series of experimentation we done. So we done this in a neonatal pap model of mice by occluding with a tourniquet the limb and see if this will create an effect on the intestine. We done it with occlusions of 5 minutes and reperfusion of 5 minutes at a very high pressure, and we're occluding that extremity. We use a very well-known model in just born pap mice, and we found this to be like almost a magic bullet. So you could see here in this panel the neck, which we stimulate with our model, and then you could see how remote ischemic conditioning, or RIC I would refer in this paper, could revert completely the damage. We look at inflammation, we look at IL-6, TNF-alpha, neutrophil infiltration. As you could see in the white column, the RIC completely ablated these changes. Also, if we look at oxidative stress, again, we found a significant effect. So we gave this remote ischemic conditioning or RIC at 3 time points to see if this is happening at the beginning of the disease, like in the blue, on the green one, or when there is already established damage in the bowel. And indeed, in the beginning of the disease, as you could see here from the blue and the green dots, we had a significant effect of RIC in the disease, but we didn't when the disease is already fully established and probably already necrosis occurring. So if we look at the survival curve of these small animals, you could see that the one with the green and the blue line, there the survival curve, which was significantly different from the one with red, which is just neck. However, the one when the RIC was given late in the exposure to neck did not have an effect. And then we continue to observe these PAPs for a longer period of time, and the curves obviously diverge between neck and the one receiving RIC as an early phenomenon in the first few days. Our neck exposure was between day 5 and day 9, and we observed these PAPs continuously later on. And how we did that? I think we actually offset and improve the intestinal wall perfusion. We measure with a very fine Doppler ultrasound, the perfusion of the intestine in these little pap mice, and we observe a significant effect by giving RIC in the intestinal wall velocity. But then we did something even more, in depth. We look at the villi with 2-photon laser microscopy, and as you could see here, the actual we could see the perfusion down to the bottom of the villi. Now I want to show you what we measure in the bottom of the villi, and you could see the perfusion in control. You could see in neck how the flow in these small venules and arteries is delayed. There is a rolling of platelets, and you could see the effect instead of RIC, where there is a much faster and almost going back to normal recovery of the perfusion. in both in the early stage of stage 1 day or 2 days. So we quantify this phenomenon. As you could see here, there is indeed an improvement in macrovascular circulation, which we measure in this using this technique of two-photon laser microscopy, in the arterial velocity, in the diameter of the arteries, and the flow in this little arteriole at the base of the epithelium, which you could see here, and I show you in the video before. Sorry for jumping. And then we look at the villi, at a single villi. As you could see, the macrovascular was significantly improved in neck in this villi. And we measure also the distance between the perfusion at the top of the villi, which was also affected and improved by giving RIC. And this is the quantification that we have here on the right side. And then we look into the, let me launch the video. You could see that the tip of the villi, which is the yellow dots, is where most of the necrosis is happening. And you could see the difference with the flow in each villi by giving RIC, where the necrosis is not present. So we quantify this phenomenon, and we look at the reduced hypoxia at the tip of the villi, which was significantly counteracted, as you could see, from giving remote ischemic conditioning. Remember what I showed you at the beginning, that in humans, actually, this phenomenon is the opposite, because in humans, we have seen a macrovascular derangement in the intestine, and this seems to be a way to counteracting that. So what is the mechanism of action? We've done a series of experiments, and I don't want to bother you with all the inside out of the experiments we did, but this is regulated by hydrogen sulfide and nitric oxide. We proved this with knockout experiments for nitric oxide, and also with vasodilator, vasoconstrictors. And so the mechanism of action seems to be related to vasodilatation. And then we look at the brain as well, because we all know that babies may not survive from NEC, but if they do survive, may, unfortunately, almost half of them survive with some neurological disabilities. And so we wanted to see if RIC actually not only protects the intestine, but protects the brain. And we look at the brain axis in this sense. And in our initial data, which is not published yet, we found that indeed the neuroinflammation that was present, the neuroinflammation which was present during NEC is completely ablated by giving remote ischemic conditioning. And you could see here with TNF-alpha, this is in the brain, and IL-6. So the glia activation, the macroglia activation seems to be responding to this RIC maneuver. We also look at the blood-brain barrier integrity, and we could see actually that in neonatal brain, by giving RIC, we protect the barrier as well. So our paradigm is this one. Indeed, we know that after feeding, the baby may develop NEC, probably because there is an imbalance between oxygen demand and oxygen supply. And by giving this maneuver, which could be relatively simple, we could induce better macrocirculation and protect intestine. And as a consequence of that, on a direct effect, we could also protect the brain, as I showed you before. So remote ischemic conditioning of the limb seems to improve the survival, intestinal injury, inflammation, oxidative stress, but also protect the brain. So it could be a novel therapeutic action. Now, we wanted to move this into the clinical setting and try to help babies like this one. And so we've done the first in human safety study in 3 steps on babies receiving remote ischemic conditioning. These are premature babies. We gave 4 cycles of maximal ischemia. And then in the step C, we gave it in 2 consecutive days. Here we have my research fellow at that time do it for the first time in babies, in fact, as small as 800 grams. And you could see we did it in 3 steps. prove that this is safe. We did it in babies from 32, and you could see here the gestational age, the median, you could see the weight, and down to 800 grams. And this is the response in the actual arm where we did the remote ischemic conditioning, because obviously we didn't want to create any possible issue to the extremity where we give RIC, and we did it mostly in the arm. The oxygen in the phase 1, oxygen saturation, capillary refill time, pain score, and urine output remain unchanged. Then we look at again in step B, where we give it the maximum dose which was tolerated in step A. And again, we look at oxygen saturation, capillary refill time, pain score, urine output, all very good and unchanged. And then in step C, we did it the maximum amount, but we give it on 2 consecutive days. This is day 1. And again, in the same arm where we did the RIC, we didn't have any significant effect on both saturation, capillary refill time, pain score, urine output. And the same remain also for the second day, where there was no significant change. So our initial safety study, which is a pilot study, showed that oxygen saturation returned in all of them in the limb where we did it. There were no new skin lesions. There was no disappearance of grasp reflux. There was no mortality due to RIC. And the pain score, which we measure, did not have any major effect. So this was a safe maneuver. Then how do we transpose this into clinical application? And now we are designed and about to start a multi-center mask phase 2 randomized control trial. I say phase 2 because ideally we wanted to jump from safety into proven efficacy. But indeed, we need to do something more. I want to show this slide because there is only 2 trials that have been done for treatment of babies with NEC. I was involved in one of these 2 trials, and both of them, unfortunately, did not prove if drain or laparotomy was better, because none of them reach the power. So that's why we want to do a phase 2 trial, because we want to see if it's feasible doing a trial on NEC in babies having a trial of RIC in babies having NEC. And we form a fantastic consortium of 12 centers from Canada, from the United States, and from Europe. And we received funding initially from CHR, and now I'm pleased to say from Trascio Research Fund. So this will be
one more minute, Dr. Pierro.
As a multi-center collaboration. Thank you very much. And the patients will be, the care team will be blinded to what procedure is done. And then we will look at feasibility of recruiting, masking, and we look at the barrier for function. We do it on babies with confirmed medical NEC, not surgical NEC, because we want to do it in the early stages, and the primary outcome is to look at the feasibility of this trial. So in conclusion, NEC mortality remains high. And so I hope that I show you some interest and stimulate your interest on remote ischemic conditioning, because we are now translating from basic science to a clinical application, and we are quite excited that this could create a future change in the treatment of NEC. This will never happen without the help of my lab, which has been fantastic in all the research work which I show you, and also the network that we created internationally, and the people, obviously, and the charities that gave us funding for doing this trial. Thank you very much.
Great, thank you for that wonderful talk. I think that people need to put their questions into the Q&A, but some of us can't do that. So I'm going to ask Venkatesh if he wants to ask his question.
Hey, Agostino, this is Dr. Sampath. Very nice talk.
Yeah, thank you.
When you presented the talk, the first thing which came to my mind was Dennis as a body study where they cooled a few patients with NEC. So the idea seems to be the same, that you can do some therapeutic hypothermia for NEC, and that might improve outcomes. So what do you think does your approach parallel the approach that, the hypothermia approach, which also is a way of decreasing the metabolic rate and also the blood flow to the bowel?
Yeah, well, thank you very much for your question. In fact, I was in London before, so I know Dennis Sampath very well. And as a matter of fact, our safety study with the 3 steps is designed on what they did for hypothermia initially in that cohort of patients with ischemic encephalopathy. No doubt we could have an effect, and we are trying to investigate if this effect on the vascular dynamics is also the primary target, followed by an effect on inflammation. So that's our hypothesis. And I think if we manage to target this population early on, we could have a short but good window of opportunity for making this treatment. So it's a little bit like for the ischemic encephalopathy, where also there is a window of opportunity. But here, obviously, it would be important to do it before the gut become too necrotic, because at that point, obviously, remote ischemic conditioning will not alter the situation. But it can alter the situation when there is a beginning of the condition. And we look at our own experience, and about 32% of babies move from what is called stage 2 disease into stage 3, or let's say from medical to surgical neck. So that's the group that we want to target.
Thanks.
All right, thank you for that question. There's a few more questions. There's one more question in the Q&A, but in the interest of time, I think we need to move on to the next speaker. So Dr. Pierro, if you can just answer the question in the Q&A box, that'd be great.
Thank you very much, Cynthia. Thanks. Bye-bye.
And I'd like to introduce our next speaker, Nicholas Embleton. He's going to be talking about the role of nutrition and lessons learned from bionutrient trials. Dr. Embleton is a consultant neonatal pediatrician at the, I'm not sure what RVI stands for, Royal Vascular Institute or something. Thank you. In Newcastle, in the United Kingdom. His program of research includes gut function and nutrition in preterm infants, but also growth and metabolic outcome in later life, reproductive loss through the Butterfly Project, and movement analysis, brain development, and cerebral palsy. So I would say he's kind of a jack of all trades. That sounds great. He's also one of the lead investigators of the ELFIN randomized controlled trial to evaluate supplemental lactoferrin on neonatal outcomes. Dr. Embleton, thank you.
So thank you very much, Cynthia, and thank you to the NEC Society for inviting me. I might be a jack of all trades. I'm certainly a master of none. I've not done any clever work like the basic scientists who've been speaking before me. Just a few conflicts, research from governmental and industry sources, but no specific conflicts to declare. And just to quickly start off with my view of nutrition, of course, we think about all the nutrients that are important when we're managing these fragile babies. But of course, we've become very aware of these functional components or bionutrients over the last 20 years or so, so particularly oligosaccharides and growth factors. The next element of nutrition, the microbial aspects, clearly these are very strongly associated with breast milk, but also the NICU environment and the role of probiotics. And then the last bit will be the technical socio-behavioral aspects, which are very important, obviously, for parental involvement, but also have aspects of sensory and taste and these sorts of issues. And when I first started as a neonatologist 30 years ago, I thought of milk really just as a food substance. We tipped it into this great big long tube called the gut. It sucked out the fluid and nutrients and produced waste product at the other end. But what we've learned in the last 20 years from all this amazing basic work is the complexity of gut function, the relationship with the immune system in preterm babies. And people are now starting, obviously, to study the various metabolites that are associated and produced in the gut, as well as the microbes, the endocrine aspects, and also the interactions with the immune system. And there's really been a paradigm shift in thinking about breast milk as some sort of biochemical signaling pathway. Okay, sorry, my slides are just skipping a bit there. So what's going on in the gut? This is a very kind of high level view, but of course there's this continual interaction between the microbial communities and the nutrients and the metabolites. And as we've heard earlier on, these microbes interact and potentially affect the epithelial and the mucus layer, and they may even translocate through into the lamina propria. And so do the nutrients and metabolites. These are likely to act at a whole series of different levels, again on the epithelium, the tight junctions, into the lamina propria. And there's also going to be systemic transport of metabolites through to the brain. And this all needs to take place in the context of the matrix of milk. So depending on what sort of milk we're feeding the baby or the pH, those are likely to affect the interactions. Now, the different types of foods that we feed baby impact directly on gut microbial ecology. So whether you are breastfed or formula fed, you receive different types of protein, and therefore different peptides appear in the gut. We know that lactoferrin does have a direct impact on the microbial ecology. Oligosaccharides have been shown to have this very directly, but also fatty acids and carbohydrates. And then in turn, the gut microbial communities produce a whole range of compounds and molecules that might be important. So particularly short chain fatty acids important for colonic function, but amino acids and peptides that might have nutritional value, choline and bile acids may have signaling potential. And then also a whole range of anti and pro-inflammatory effects and signaling molecules that may impact on the brain. So this is a very complex system, and it's going to be very difficult for us to study it. Now, what are the bionutrients we're talking about? I mean, clearly, almost all of these appear in breast milk, and that's where all the new science and the ideas are coming from. And these bionutrients might be hormones such as IGF-1 or insulin. They could be enzymes, particularly proteins and studies. So say obviously lactoferrin, but also osteopontin and catecholine components, specific amino acids, nucleotides, immunoglobulins, particularly IgA, fatty acids. So we know that GHA and arachidonic acid have signaling activities, carbohydrates. We're not really going to consider microbes and cells as being bionutrients, but clearly they're integral to this system. And then lastly, the what you might call multi-component products that are clearly not breast milk, but may be considered to be sort of multi-component bionutrients, such as bovine colostrum, which is now being tested in clinical trials, and milk fat globule membrane. So there's really a huge number of molecules and proteins that we should be thinking about. And many of us are starting to sort of hope, I suppose, that bionutrients might really be the holy grail of nutrition. Our focus here is on gut health, but we shouldn't forget that many of these bionutrients may have beneficial impacts on the brain. But what we're really trying to do here is hope that we might be able to decrease the risk of NEC, particularly surgical NEC, prevent death, but also avoiding NEC, and therefore avoiding the cytokine storm and avoiding brain damage. So overall, what we're trying to do really here is improve gut health. And measuring gut health is really pretty complicated and complex. What sort of markers should we use in preterm babies? And as Cami and Venkatesh and Agostino have been showing you, there is some amazing kind of preclinical and animal studies, and there are a few sort of mechanistic studies. And they're using and exploring lots of different mechanisms and biomarkers, but really none of these RCTs are powered to detect differences in functional outcomes, particularly disease. So here's my very high level simplistic view of how the bionutrients might be acting. They might be acting within the lumen of the gut. So for example, lactoferrin binds iron and may reduce antioxidant toxicity. They may impact on digestive function and motility, and they may impact obviously on the microbial community structures. Some of the bionutrients, a lot of data show they impact on intestinal growth and differentiation. They may well have anti-inflammatory effects. Good evidence, as Misty was talking about, for the role of an impact on mucus layers and tight junctions. And some of these bionutrients may well translocate into the lamina propria and impact on immune function and, for example, IgA production. And then thinking another step out, nutrients may impact on the intestinal blood flows. There is some work and a couple of clinical trials looking at arginine. They may have signaling roles further afield, and they're likely to impact, obviously, on systemic immune function. So a very complex system and lots of different mechanisms that we might be interested in. But note that just because a bionutrient ends up in the gut, it doesn't necessarily mean that it's going to have functional activity. Because if you go and collect stool from babies, you will find intact IgA and intact lactoferrin, and therefore it's very unlikely to have had a huge impact on gut function. Now, interestingly, many of these proteins that we're looking at have ancient origins. And this is because infection really has been one of the driving forces of evolution. And if you look at taxonomy of evolutionary development in animals and then into mammals, you'll see that many of these proteins appeared well before humans ever appeared. I mean, lactoferrin is present in worms. We know from a whole series of studies that the ability to create lactose from alpha-lactalbumin appeared at least 250 million years ago. And again, there's evidence from reptiles that oligosaccharides probably evolved before lactose did. And of course, many of these bionutrients are present in amniotic fluid. So these are ancient proteins that are likely to have huge impacts on microbial aspects. And this is why they're so important when we're studying and thinking about NEC. Now, my very simplistic view of NEC is just to think about the interactions between the micronutrients, the nutrients, the microbes, and the immune system. And we know that this relationship in healthy breastfed infants is well balanced, and the relationship between these three aspects is likely to change over the life course. Particularly in preterm babies, this is a very disturbed interaction. The babies are often not born by vaginal delivery. They live in very sterile, clean environments. Their skin is broken. They have bits of plastic inserted into them. The breast milk we give them has been affected in lots of different ways, and then they're exposed to antibiotics. So it's hardly a surprise that this interaction in preterm babies is so easily disturbed. And you can, on a very simple level, I think of NEC as a breakdown between these three different components. Now, whilst the focus of this webinar today is on NEC, you could think of NEC as this tip of the iceberg. And lurking below the surface really are all the different aspects of gut health that we're currently struggling really to define and measure. But this may have impacts clearly on sepsis or subclinical infection and inflammation, may affect feed tolerance, nutrient assimilation, and therefore growth, impact on antibiotic exposures, and also long-term metabolic outcomes. So we shouldn't lose sight of really what we're studying is trying to improve gut health in babies rather than simply avoid a dichotomous outcome like NEC. So when we want to think about outcomes, if we think of it like a PICO format, what sort of outcomes should we be trying to measure? Well, the real problem here is that NEC is rare. You're going to need very large trials. Biomarkers, whether they're fatty acid binding proteins or cytokines, they're not really outcomes, and we don't know how they're going to relate to clinical practice. As we mentioned, NEC isn't a single disease. NEC that is driven by a vascular problem is unlikely to be benefited perhaps by lactoferrin. Composite outcomes, so for example, including death, NEC and sepsis as an outcome, is very problematic in clinical trials. And finally, just to highlight that NEC and intestinal perforation are really very poorly discriminated. And my colleague, Dr. Barrington, has recently published on this, showing that many of these cases are not as robustly diagnosed in clinical practice. So when you're using big databases of clinical outcomes, they may not adequately distinguish between NEC and intestinal perforation. And furthermore, NEC stage 2 is not a robust diagnosis. Now, NEC is not a very helpful term. I mean, it's the end result of perhaps multiple different pathologies, and really it's a pathological term that we're using for a clinical syndrome, and that I think makes it particularly unhelpful. If you look at this scheme, you can ask yourself the question, at which point does this baby have unequivocal NEC? We can't look at the gut until we take the baby for surgery. So how do we know really whether this baby definitely had NEC? Is it possible that this is a baby presenting with rapid onset sepsis that's giving the appearance of NEC? Or could this be an intestinal perforation that was missed and the baby has now developed peritonitis and bowel inflammation as a secondary phenomena? And all of us in the meeting today could spend hours and days arguing over which of these different baby combinations of x-rays and distended abdomens and gastric aspirates of different colors and volumes, all of us could argue endlessly whether this was definitely stage 2 NEC or not. And then there's a lot of variability between neonatal units depending on how you practice. So your attitudes and behaviors to stopping feeds and getting an x-ray and deciding that that's pneumatosis will impact your NEC rate. Variation in how willing you are to take a baby to surgery, and then whether you interpret the histology correctly, or even if you check the histology when you're writing the discharge letter. And even when we come to post-mortem review, it's clear that not everybody reviews their detailed histology and surgical findings accurately. Now, running trials in a busy neonatal unit is incredibly hard for any of you who've done it. Here you've got a terribly sick baby with lots of clinical pressures. And then somehow on top of that, we've got to find time to screen and consent, to speak to parents, make sure they understand, find out who's going to collect the data, check it, and what you do with incomplete data. What happens when the unit's too busy to recruit the babies? Have you got a research team available 24 hours a day? These are all the complexities all of us are facing when you're trying to do clinical research. Now, as Cynthia said, we did this trial on lactoferrin, which is at very high levels in human colostrum, and even in mature human breast milk, much higher than in cows. As I say, this is an ancient protein, very highly conserved and present in lots of mammals, but crucially, the structure does vary between different species. We know it's likely to work through a whole series of different mechanisms. And it's present in lots of different body secretions. And it may well act on bacteria, viruses, or fungi, or modify host immune responses, act on the epithelium, act on iron sequestration or prevention of biofilm. So lots of potential, and there was a lot of hope that lactoferrin really might be beneficial. And so we planned this large elephant trial where we recruited 2,200 babies. And over a period of about 3 or 4 years of recruitment, we successfully showed absolutely no difference between the lactoferrin group and the placebo group for a primary outcome of late onset sepsis. And there was also no difference in microbiologically confirmed sepsis or NEC. Now that came, not surprising, as a huge disappointment given the effort we put into planning this trial. So running a trial of a bionutrient like this is incredibly complex. You need really rigorous quality control and assurance. All of this is watched very carefully by the regulatory authorities. You need to make sure that you account with various legal frameworks, that you can account for the different products that we're using and disposing of. You need to prove it's lactoferrin. So someone might sell you the lactoferrin, but how do you know that really what you've been given is lactoferrin? Lactoferrin purity is going to vary between batches. Iron saturation and binding perhaps affects bioactivity, and the storage and the temperature and the duration of this also changes the potential bioactivity. You need to get something that's incredibly safe. Our lactoferrin came from New Zealand because it hasn't had most of these horrific looking animal diseases. But then you still have to think about pesticide contamination, yeasts and mold, and be aware that even when you pasteurize, you may not inactivate endotoxins in that process. There's a paper from Bo Lonadal that came out at the end of
Dr.
last
Embleton,
year.
this is your 1-minute warning.
Oh, thank you. So there are 4 different plots here, endotoxin levels, proteomics, from 8 different types of lactoferrin, and you can see that all of these vary considerably between the substances. Masking and making it up was very difficult. We spent a lot of time trying to find a product that we could use as the control. This again took a lot of work, producing it in a way that you can actually draw it up in clinical practice. And then when you work across 134 hospitals with different syringes and nasogastric tubes, you introduce further complexities. And then all of this is monitored. Temperature on the neonatal unit may impact on storage conditions. It took us 10 years from thinking this idea to actually doing it, and £3 million. These are very expensive. So bionutrients are complex interventions. There are lots of different interacting elements that may be positive or negative. The trials need to be sensitive to context, depending on the milk matrix and the types of milk you're using. You need to standardize the intervention given the different commercial sources, recognize that there's a lag because nutrients are not drugs, and recognize the complexity. So in the gold standard trial, pharma-type trial, there are going to be lots of issues around the population. You can't predict at randomization which babies will be breastfed or formula fed. And then the intervention, as I say, lots of issues around blinding and masking and the acceptability from our parents. And then thinking about the outcomes, you need very large trials if you're going to study NEC, and you need to consider what you do in terms of analysis if you have a baby who is intended to get the treatment but doesn't actually receive it. So I've rushed through all of that. So my final slide here really is just to emphasize how important it is. NEC kills more babies than all childhood leukemia and lymphoma combined. But nutritional strategies offer many opportunities, particularly bionutrients. And I really believe that there are exciting times ahead, both for the interest now in oligosaccharides, but also growth factors and advances in biotechnology. There are lots of very good preclinical and pilot studies, but we really need more, I think, before we launch into our next series of studies. We need great product quality control and assurance. We need robust and accurate tools to diagnose NEC. But then we're going to have to conduct very large collaborative trials, hopefully with nested mechanistic studies to understand it, but also using bionutrients that are going to be effective when they're added to different gut milk matrix in the gut. But finally, also, it may be that better understanding bionutrients may help us select donor milk that has a more optimal profile. So for example, in the composition of human milk oligosaccharides that are more effective in reducing NEC. And I'm going to finish there. I'm sorry if I had to rush, but such is life today. So thank you very much again.
Thank you, Dr. Embleton. That was a great talk too. Sorry, we needed to squeeze you in. Does anybody have a question on this last talk? Yes, so Christopher Stewart has a question for you. What do you think is the next ELFIN trial for preterm infants with NEC as primary outcome?
Yeah, thanks, Chris. Well, I think we're all tempted to sort of think that something quotes a bit more simple, like an oligosaccharide, might be a great idea given the sort of data that's already coming out there. But I think my point would be we need to be cautious. Actually, we can't go running £3 million 2,000 baby trials without having thought really, really carefully about the design. So at the minute, I wouldn't favour anybody rushing into a large bionutrient trial unless you've got really good evidence that the intervention you're using is likely to be effective, and you really decided that that's the most important one. So we're going to have a collaborative approach here to think carefully, I think, before we launch into the next trial. Great.
Let's see, is there another question here? Yes, here's another question. Great talk. Would be a difference if it is human lactoferrin versus bovine lactoferrin?
Yeah, good question. I mean, there have been some studies on human lactoferrin, but not surprisingly, it's going to be much more expensive. Now, that doesn't necessarily preclude it, but there could be supply issues with a human-derived product if you're doing a trial of 2,500. And there have been concerns about the recombinant human lactoferrin in some of the adult trials. So again, there could be safety issues. The temptation is to think that a recombinant product might be safer, but we don't really know that. I mean, the advantage of using something from a cow milk is that it's pretty cheap and widely available, but I agree, humanized products might be preferential.
And one last question, which is actually in the chat box, and that is there an animal model that you trust?
We don't do animal work, so I'm not going to cast any aspersions on anybody else's models, but it's clear that most animal models, the type of neck that they see is not really what we see in preterm babies. So I think they're great for obviously the preclinical workup, but we need to go another step beyond an animal model, I think, before we choose the next bionutrient for a big clinical trial.
Do any of the other panelists who are still on want to answer that question also?
Nick, I have to answer that question now, right? So I agree. I think no animal model is perfect, but I would argue that it's the best one we have. And I think the Android model, some of the stuff that Misty is doing is actually very good as a sort of step between, but there are certain fundamental questions which can be only answered in the animal model. And if you want to test new drug therapies, safety, and you still have to go that route before jumping into human studies. So I think there is a value. We all understand the limitations of the model. Now, I would, I mean, I do clinical work too, so I can tell you that what we call as NEC, as you pointed out, is not even one disease. So even in the clinical side, there's a variation in the phenotype. So I think understanding the limitations of the model is more important, and understanding the limitations of our definition itself on the clinical side is also important, I think.
Yeah.
Anybody else want to comment on that?
I will. This is Misty. I agree with Dr. Sampath as well. I think, you know, one of the things is we all cherish our babies, right? And we can't run out and test every little idea that we get as scientists on real live humans. And so I think the animal models, they are what they are, and they all have limitations, just as neck on a chip and the Android models do as well. So I think just keeping that in mind, we really can't get anything through the FDA, any drugs or anything like that, without animal model testing. So we have to keep trying.
And with that, I'd like to turn it back over to Jennifer to conclude this webinar. Thank you. Thank you to all our speakers.
Thank you. Truly, shout out to all of the speakers here today. Your work is phenomenal and so exciting, and really gives us hope that we are building a world without NEC, that it's going to take a long time, but we can get there, and that there is hope. So thank you for your extraordinary dedication to patients, families, and to building a world without this really devastating disease. I'd like to invite you to register for our remaining 2 webinars that we're hosting in partnership with Pediatric Research. We have one coming up on February 14th and another on February 18th. You do need to register for these individually, so they're separate registrations. You can visit nexsociety.org to access that registration. I also want to give you a heads up that May is NEC Awareness Month. May 17th is NEC Awareness Day. So if you are interested in checking out our shop, you can visit nexsociety.org. I know you saw on Misty's slides a lot of her team members were wearing NEC Society gear, so you can get that online at necsociety.org. And again, just want to recognize and honor sweet, beautiful baby Tom, and thank you for carrying his memory in your hearts as we go on and build a world without necrotized enterocolitis. Thank you so much again, everyone, and we hope to see you again soon. Thanks.